Welcome to today's Green Talk. I am super excited to be introducing Dr. Luke Miller. Luke Miller could actually have his own department called E&E, which would be Electronics and Ecology. As a biologist, he has sort of unparalleled knowledge in designing circuits and making devices that work outdoors in the ocean. He did his PhD at Stanford University at Hopkins Marine Lab in Monterey, a university with its own private bit of intertidal shoreline. And he did his undergrad at UC Santa Barbara. Luke Miller teaches biostatistics and courses in ecology and invertebrates in biological sciences here at San Jose State. And his talk actually segues very nicely with last week's presentation on habitat affecting wave structure. Luke Miller has built software that looks at waves and temperature and things in real time, but on a very microspatial scale, second to second at the scale of organisms like snails. So I'd like you to give him a warm welcome. Thanks, Luke.
Thank you. So I am trained as a biologist. I came through my PhD lab, a lab that was really sort of focused on using physical principles and engineering principles to look at how biological organisms deal with their environment, with the weather, the wind, the waves, all of those things.
We just should check your microphone. Is that on?
Green light.
Thanks. Good.
So I'm interested in how organisms, particularly that are living out on the seashore, deal with, basically survive in their fairly highly variable environment. Those waves that are crashing at high tide, they're heating up at low tide, trying to feed, trying to reproduce, trying to hold on even. I'll put this in the broader context of how climate change is potentially impacting our entire world. This is a figure here at the top from the most recent Intergovernmental Panel on Climate Change report back in 2014, summarizing projections on how much surface air temperatures are likely to rise by the end of the 21st century relative to what they were at the end of the 20th century. So we've got two different emission scenarios here, one bad, one worse. And in general, in most places on the globe, we expect to see increases in temperature as time goes on. And this is really about average temperature in this case. So we expect an upward trend in a lot of places. Layered on top of that, though, is the fact that we're also expecting, based on global climate circulation models, that things like heat waves will become more frequent, more intense, longer durations as well. And those are things that can potentially really start to remove individual organisms from their environment via killing them on a scale that might start to matter for whole populations, whole areas of the coast or whole areas of the forest or wherever. So this figure here is showing us the expected increase, or one projection on increases in June, July, and August extreme temperature events. And this is the percentage of years here in color where we expect by the middle of this century that the percentage of years in the middle of this century that we expect to have June, July, and August heat waves that exceed what normal heat waves used to be at the end of the 20th century. And so here along the west coast of the US, we expect to see somewhere, let's say, between 60 and 80% of the summers that we experience by about the year 2050 will have heat waves that were substantially worse than what we ever saw back from the 1960s through the mid-2000s. And so what that's doing is really adding on variation around that change in mean temperatures that we're expecting in the future. And it's that variation, particularly at the high end, that is potentially what's actually killing organisms, or at least impacting their energetics and allowing them to not really grow and thrive as much as they would like. So I work in particular on rocky shorelines. So this is an aerial view of the Hopkins Marine Station down in Pacific Grove, Monterey. For reference, down here in the corner, the lower left corner, is the Monterey Bay Aquarium. So it's right adjacent to the Monterey Bay Aquarium. And it's a nice little stretch of coastline that's fairly protected from human impacts, except by me, and gets a lot of waves coming from the north. You get sunny places, shady places, you get wave-exposed places, you get wave-protected places, and there's lots of animals and plants or algae that are living on these shores. Now, really, the habitat that's living in this intertidal zone between the high tides and the low tides, it's really shaped in large part by what the tide cycle is doing. So I've got a couple of weeks of high and low tides mapped out here for that site in Monterey. And what you see is a couple of times a day, and literally a couple, 2 times a day, we have a pair of high tides. Those are balanced with a set of low tides that also happen twice a day. And around here on the west coast, we see this mixture of a higher high tide at one part of the day and then a not quite as high tide later in the day, and a lower low tide at some point in the day, and a not quite as low tide later in the day. And the amplitude of that is changing constantly through time, or cycling through time, I should say, between these bounds. And so here we've got tide height on the y-axis, and you can see there's a range of about, 2 meters or so in Monterey. And so there's a stretch of shoreline that you go out there that is alternately being exposed to the air and then submerged by the water twice a day. So at high tide, you've got waves potentially breaking over this shoreline. And all the animals and the plants, the algae that are living there, essentially have to sit here and hold on, not get ripped off those rocks. They're also often trying to do all of their feeding under these conditions, whether it's by grabbing onto neighbors and drilling through them, or filtering water, filtering food out of the water, or just doing photosynthesis as a piece of algae. You do need to be wet in order to do that. And so this, at high tide, is when a lot of the biological activity is really happening. At low tide, though, the water drops, and all of these marine organisms that do need salt water to survive, they need to be submerged once in a while to survive, are exposed to terrestrial conditions. So suddenly they're sitting in the sun, potentially. The wind is blowing over them. They're drying out. They're potentially heating up, or at night, they're potentially getting quite cold as well. Because really that ocean provides this nice, relatively stable temperature environment for them when it's over the top of them. But when it goes away, they're exposed to really the vagaries of what the terrestrial weather does. So we've got a lot of different critters living here. We've got some pieces of algae, the green stuff or dark green stuff hanging off. We've got sea stars, which you ought to be familiar with. We've got barnacles, we've got mussels. All of these things, huge amounts of biomass, living material attached to these rocks and making a living here. And I'm really interested in how these things do that, how they survive, how they cope with particularly high temperatures. And so really we want, as biologists, to be able to answer the question of how to predict how these sorts of communities, these mixtures of all these different animals and algae, are going to cope with future climate change. And the way we make projections, of course, is to build models that we can run into the future. And there are, of course, basic statistical models, and then there are mechanistic models that we can put together to try to make these projections. And I really like to focus, in particular, on mechanistic models. And so mechanistic models, this is going from first principles physics concepts in some cases, are really built, when you're talking about biological organisms, around the idea that we need to know how the biology interacts with the basic physics of some organism. So if we're heating a piece of algae up, if we're heating a snail up, when does it start to die? When is it perhaps actually running at its fastest? Because all these things are generally cold-blooded, they're ectotherms. And so as their temperature changes, or as the temperature of the environment changes, their temperature changes, and they start moving faster if it's hotter, and they move slower if it's colder. So these mechanistic models need to be parameterized with biological limits in them. And that sort of information can come from lab experiments, and so I'll talk a little bit about some lab experiments that we do to generate these parameters, and also field experiments. And field experiments are nice because you get out there in the wildly variable natural environment and try to characterize really what the limits are, or what the range of variation is under these very complicated natural conditions. And ultimately, all of that feeding back into models that we can use to make predictions about who lives, who dies, who prospers, who fails in the future. So just as an example of mechanistic models that I've used in the past, I've developed heat budget models for individual snails living on the seashore. So this is a snail that in reality is slightly larger than a pea out of a pea pod. But they live on the rocks and they crawl around at low tide, or sorry, at high tide grazing algae. And at low tide, they sit like this. They close themselves up inside the shell to try to hold on to the water that they have. And they can be exposed to the sun and either heat up or cool down based on the various basic inputs that you have. So you can have conduction from whatever the rock underneath them is doing. These are living on big granite boulders, and so that rock doesn't heat up very quickly. It also doesn't cool down very quickly. You can have long-wave infrared heat exchange with the sky, with the surroundings, with the physical surroundings below them. You can have convection from the air, so air temperature can definitely impact the body temperature of these animals. It can cool them off, or it can heat them up. And then shortwave radiation from the sun during the day is obviously a major input into the heat budget of these things. So the goal here is to be able to take basically a bunch of weather data and start to predict animal temperatures from that, and then know something about whether those temperatures that the animals are experiencing are going outside of the boundaries of what they can actually withstand and survive physiologically. So the model, once you start pumping weather data into it, starts to predict, in this case 10-minute time steps, the body temperature of an individual snail over the course of a day, through the middle of the night, into the morning as the next day comes and the sun is rising again. And you can baseline or establish baselines and test how accurate these models are by putting out little models of live snails or fake snails, measuring temperatures in the field, and then looking at how well your model does in terms of lining up what it thinks the temperature of any one of these snails ought to be based on the weather conditions versus the temperature that you actually measure in these snails. And so we get plots that look like this, which to an engineer should look ugly and a lot of variation here. But to me as a biologist, if I'm 2 or 3 or 4 degrees off, I feel pretty good about that model actually. That says that for all of the wild variation in terms of the wind speed and the boundary layer thickness over the rock surface and the color of the animal impacting how much heat it absorbs and all that sort of stuff, given all those different inputs, we actually do reasonably well in terms of matching up our measured and predicted body temperatures. And so then we can start to generate time series over longer periods of what we think body temperatures ought to be for these animals living on some certain piece of rock on the shoreline, and how often they start to hit particularly high temperatures here. And for me as a biologist, a temperature above about 30 degrees Celsius is where I start to say, yeah, that's getting hot. That's taking one of these marine animals and getting it above a temperature where it's starting to feel the heat stress, the physiological stress of being that warm. And so through time, you can start to look at how frequently they should actually experience those particular high temperature events. Now, as I said, lab experiments are useful for creating some of the parameters, particularly the sort of physiological or biological parameters of how these animals respond to different conditions. So this is just a little video of a lab mesocosm study I did a couple of years ago. Inside these little buckets here, we have seawater, we have snails and mussels and crabs living inside here. And we're simulating low tide right now, so the water level is dropped. At high tide, the water level will be high, submerging everything inside the buckets here. And then we're heating them up individually with these heat lamps. In some of the situations, we also have controls that aren't being heated up, in order to look at how the animals in there, how quickly they're feeding, where they're spending all their time. Are they hunkered down underneath some shady spot, or are they out in the open under a heat lamp? How much are they growing over the course of several weeks? Those are the sorts of things where we can track temperatures in these conditions, we can track animal performance in these conditions, and use those to start to better our models in terms of predicting out in the field how that animal ought to be performing. So just some temperature data from one of these experiments. In blue, we have the variation in ambient temperature. So this is seawater temperature at high tide. This is air temperature at low tide. And then in red, we have the effect of those heat lamps, and we also had seawater heaters as well for high tide conditions. So we're, in this case, just trying to raise the average temperatures of half of our mesocosms in order to get an idea of how the animals are responding to that over the course of several weeks. We run these for a month at a time. So we have this group of 3 organisms here: a crab that likes to eat snails, a snail that likes to eat mussels, and mostly we're sort of focusing on how this snail performs, how many mussels it eats, how much it grows, if there's some impact of just having crabs around, essentially peeing into the water and scaring these snails, and also changing the temperature on them. And so one of our mesocosms will have crabs down in the bottom, it will have snails and mussels up at the top, snails eating the mussels, mussels being very unhappy, both in the heat and also because they're being eaten. And then this crab down here trying hard to get at the snails but never actually reaching them. And so what we see under these sorts of tests is we can do things like measure the total growth, in this case of the snails, under those different combinations of conditions. So with crab chemical cues, without crab chemical cues, under ambient temperatures, under warming temperatures, and sort of relevant to what we're talking about today, it is this case, the difference between our blue line up top and our red line down below seems to be the impact of just having a small 2 to 3 degrees Celsius temperature rise on the performance of these snails. So they are still eating, they're consuming mussels, they're generating new shell mass and new tissue mass, but they also tend to burn energy more quickly. They're essentially more wasteful when they are warmer. And so we see lower overall growth, but we also see lower overall efficiency as well in terms of calories coming in and calories being turned into, or new calories worth of tissue and shell. So then I want to turn to most of the bulk of today's talk, which is talking about doing field studies. And we're going to turn to really looking at mussels. So if you're not familiar with mussels, I mean, hopefully most of the people in this room have at least sort of seen them served in a restaurant somewhere. Mussels are effectively just rocks with food inside of them. So they have a hard outer shell of calcium carbonate, this black stuff here. When you cut them open, what you've got is a bunch of tissue inside. Mostly, actually, a lot of gonads, all this pink stuff here. That's primarily what you're eating when you're eating mussels, steamed mussels. So you want to get good, ripe, fat, ready to reproduce mussels on your dinner plate. They make their home on the seashore, again, being bashed by these waves. And the way that they hold on is they actually secrete these little thin proteinaceous threads, byssal threads they're called, that they glue to whatever hard surface they can find, whether that's the rock surface or, in this case, their neighbors. And they build these aggregate, these groups of mussels that are all sort of glued to each other, but also to the rock underneath. They make these beds of mussels. So at low tide, when the water has gone away, they tend to clam up. They close up the two halves of those shells. They're trying to conserve whatever water they have in case they get heated up by the sun and start desiccating and drying out, and that's bad news for them. Also, there's no water washing over them right now, so there's no way for them to filter feed, to capture food out of the water. So they just clam up and hang out for a couple of hours. During the course of this, they go from doing aerobic respiration to anaerobic respiration. Because they've sealed themselves off from the outside air, they use up what oxygen they have in their internal fluids and essentially become anaerobic or anoxic during that time. So that's a little bit energetically wasteful. And if they're particularly warm during those anaerobic periods, they're really wasting a lot of energy. Their internal machinery is running faster, but it's running anaerobically, and they're just burning calories. So at high tide then, what the mussels will do is they will actually crack open those two halves of the shell, and they will start pulling seawater in order to, A, reoxygenate themselves, but also, B, start to filter little bits of plankton out of the water, and that's what they feed on. And so during high tide, they're hopefully happy, submerged, feeding lots and lots. But as I've sort of alluded to, these mussels form these big beds, interconnected individuals, and they cover a lot of real estate in the intertidal zone. They sort of live in the middle of the intertidal zone. They don't go all the way to the top where it rarely gets splashed. They don't go all the way to the subtidal or underwater, permanently underwater zone. They just live in that middle range where they're twice a day coming out of the water, twice a day going into the water. And they are then food for a lot of other organisms. They are food for humans in some cases. Some of you probably have relatives that like to go fishing at the seashore, and they will crack open mussels and use those as bait on their fishing lines. They also provide sort of a home and refuge for all sorts of other little critters that live inside the spaces between these mussels. So I want to really start to better parameterize mussel or models that tell us how these mussels which are an important member of this community, are going to cope with future warming. Now, what folks have done for many years is actually try to measure temperatures inside mussel beds out in the field. The way this is typically done is to take a commercial temperature data logger like this Tidbit or this iButton here on the upper right and embed that in something like an epoxy mussel. So this is essentially a biomimetic temperature logger that can be glued down inside a regular mussel bed. These are fairly small mussels relative to the logger, but mussels do get as big as those temperature loggers. And you can put these out here and have them sit there for months at a time and record a temperature every 5, 10, 20 minutes or so, however long you want this to run before it fills up the memory. And you get these nice tracks, time series, of what temperatures were. You sprinkle a couple of these across a mussel bed and go to different sites up and down the coast, and you can build essentially a library of data in terms of how hot and how cold at least the biomimetic mussels are getting. And hopefully that is a good analogy to what the temperature of the live mussels is. So folks have been doing that for a lot of years, putting out those biomimetic mussels up and down the coast. So this is a picture of the West Coast up to Washington at the upper tip here, down into Southern California at the lower end of this plot on the right. And the heat map here is essentially a series of, average daily maximum temperature for the mussel loggers through time on the x-axis for different sites up and down the coast on the y-axis. And the color here is that average daily maximum temperature. And so where we are seeing red and yellow blotches of color are sites along the coast where mussel beds seem to be getting particularly hot and maybe getting really thermally stressed and thus physiologically stressed. And also probably occasionally, if it's turning red here, probably actually killing live mussels in those beds. And so those are areas where we perhaps start to worry about how they're going to do in the future if we go beyond what this natural current day temperature variation is already like. Now that's at large scales. We're talking hundreds of kilometers along the coast here, in some cases between these sites. We see a fair amount of variation. If you go down in scale, spatial scale, to the scale of a single mussel bed, you see almost an equivalent amount of temperature variation across the space of a meter or two. So this is a plot from a paper a couple of years ago, research done at Hopkins Marine Station. The x and y axes here, the bottom plane here is distance along the shoreline. So we're just looking at a few meters, basically 1.5 meters in the east and north and west and south directions. The colors here are meant to represent the height of this mussel bed above the zero tide level. So red colors here, these are higher portions on the shore. They should be out of the water more often. They should spend more time in the sun heating up as well. And then the individual dots here are the locations in space of those biomimetic temperature loggers. And the numbers here alongside them are the average daily maximum temperature for that individual biomimetic logger. And I've just pointed out two in particular here that are separated by about 1 meter on the ground that are, in one case, only reaching about 22 degrees Celsius on average every day. And the neighbor just a few body lengths away is reaching temperatures above 30 on average every day, which means in some cases it's probably hitting 35, 37, 38 degrees Celsius on certain days. So there's a whole lot of potential variation in temperature just at the scale of a few body lengths for these things, even though they're sitting on a fairly horizontal bench in the mid-intertidal, being splashed by the same waves, being exposed to the same sun. And this range of variation is actually a little bit troubling to someone like me because it means we actually have to do a better job of characterizing how much of that variation there is at that small scale before we can feed it into our models and start making projections about the entire coastline. And again, these are all data based on biomimetic loggers. We don't actually have good temperature records for live mussels in these same beds. But that's sort of what I'm going to talk to you about today, is trying to get those temperature records for live mussels. So we'll talk about how we actually monitor these things in the field. We'll look a bit about temperature differences between mussels sitting within the same bed versus different places on the same shoreline, higher on the shore, lower on the shore. And we'll look at gaping behavior, that is the opening and closing of the shell valves. because when these things close up at low tide and they're sitting out in the air, as I said, they're going anaerobic. They're reducing their desiccation, meaning they're not losing a bunch of water. That's good for them. But it's also an indication of sort of when they feel like it's safe to actually open up, it's safe to start feeding, those sorts of things. And so we want to monitor gaping behavior as well. So the EEs in the audience should all clutch their hearts and go, oh my God, that is ugly. What moron did that? I did that. The beauty of being alive in this day and age is that you can sit down with free software in front of a computer and bodge together something like this that is a custom temperature, or no, sorry, just a custom data logger that we're going to use to track muscle behavior and muscle temperatures in the field. So we put these together, have a fab shop send us a bunch of printed circuit boards, assemble them in the lab, and what we've got then is a nice little cheap $50 to $60 data logger that's based off of an Atmel Atmega. This is the same sort of brain that's in an Arduino if you've played with those. We can write our data to a micro SD card, so we've got essentially unlimited memory capacity here. We've got a real-time clock, so we can sync all these things up and know what time of day, whether it was high or low tide, we were measuring the readings we were taking. We've got thermocoupled A to D converters so that we can turn temperatures from muscles into digital signals. We'll talk about some of the other sensors as well. All of this gets powered off of a set of 4 AA batteries so that we can power them cheaply. They'll run for about 2 weeks on that power supply, and then we can go out and replace them. As far as actual sensors that we're putting on the muscles, since that was just the brain board I was showing you, we built little, essentially little carrier daughter boards here with individual sensors on them. I end up waterproofing them with a little bit of adhesive-lined shrink tubing. So I abuse stuff that I get from the hardware store and from the McMaster car catalog in order to make sort of marine-grade data logging equipment. We package all of that in nice little waterproof boxes. And these are capable of actually being underwater for hours at a time. We have little bulkheads so we can run these sensor wires out of them. And then each board here is actually reading data, sensor data from 2 different muscles. So we'll pack 3 boards into one of these boxes, and we can take all of this and bolt it out in the intertidal. So then I want to talk a little bit about what we're actually measuring, in this case with live muscles. We start off with a temperature sensor. So we use thermocouples because they're tiny. They're not terribly robust, but as long as we strap the wire down, they sort of tend to last in the waves. And we actually drill a little hole through the shell of the muscle so that we can insert the tip of the thermocouple in there, glue it back up so we're not losing water out of that hole, and we can get an internal body temperature for these muscles. And we're essentially just sort of jabbing the muscle in the gonad. And they seem to be okay with this. After a couple of weeks, they actually start secreting new calcium carbonate to try to cover up this little probe that we've left inside of them. And so we can get an internal body temperature. To measure that valve opening and closing, we use a Hall effect magnetic sensor, one of these Allegro units, and we will glue the Hall effect sensor to one of the 2 shell halves, and a muscle to the other, or sorry, a magnet to the other shell half. And so as the 2 valves get further apart, the magnetic field weakens, and you see that as a change in the voltage coming out of the Hall effect sensor. And we can read that signal with the onboard A to D converter for these from the Atmega, and get a reasonable picture of sort of how far open or closed this muscle is through time. So the temperature and the Hall effect data we're reading once every second because, again, we sort of have unlimited memory, so we might as well. The other thing we were interested in is whether these muscles are changing orientation, changing their body position through time. So to do that, we have one of these LSM303D 3-axis accelerometer magnetometer packages on one of these little daughter boards that we can glue to the outside of the muscle shell as well. We put it on the same side as the magnet so that the distortion in the magnetometer from that magnet is constant and we can calibrate it out. Calibrating them required sort of writing a whole little workflow in the software that I used, but we can make nice 3-axis acceleration and magnetometer data out of these things and convert that into real-world orientation. You know, is it laying down? Is it standing up? Is it pointing north? Is it pointing south? those sorts of things. And we're measuring these at 4 hertz, just a little bit faster, to also potentially see if we see a little bit of signal of waves hitting them and moving these things around, accelerating and decelerating them. So all of this stuff gets glued onto the outside of the muscle shell. We've actually got the thermocouple wire sort of covered in glue and going into the shell here as well. And in total, we can put 12 instrumented muscles, 12 wired muscles, on a board like this. So this is essentially the size of a cafeteria tray here. We've got 2 of our waterproof boxes, and then wires go into the 12 different muscles. And we've sort of spread them out here, mostly so the magnets aren't interfering with the neighboring sensors on other muscles. And then we also pack in a bunch of other live muscles here. We do all this back in the lab in a water table so that the muscles aren't being washed around by the waves, and it gives them time to attach themselves to this acrylic plate underneath and also to each other, and essentially rebuild a little muscle bed for themselves so that they're anchored to their neighbors and anchored to the bottom. And then we can pick the whole plate up and take it out into the field, drill a couple of rock anchor holes, and bolt the thing down in the intertidal. So they're now being exposed to the same natural conditions that they were when we originally plucked these mussels off of the rocks at Hopkins and brought them into the lab. So these are mostly all adult mussels. They're about 60 millimeters long or so. So they've been around for years and years, but they represent about as big a mussel as we tend to get at this field site. We take these plates. In total, we had 2 plates that had the full complement of 12 mussels on them. We had 1 additional plate that had 6 mussels on them that we put in a tide pool. And so we put these things out in the field. So this is an aerial view zoomed in on a portion of the shoreline at Hopkins Marine Station. So this is a seawater channel here, and then a lot of granite bedrock as well. And our 3 stars here represent the 3 different sites we put these plates out at. And an on-the-ground view of where we're working then. Here's one of this is our high shore plate in the foreground of the photo on the right-hand side here. Here I'm bolting down the low shore plate in the background. So they're separated horizontally by about 4 or 5 meters. It's not a whole lot. Vertically, they're separated by about 0.7 meters. And all of these plates are sitting above the mean lower low water, which is the 0 tide level. But they're sitting sort of in the middle of the tide range, which we said goes up to about 2 meters earlier. The third plate, we submerged in a high shore tide pool. And so this is a pool that never loses its water during low tide. It retains it like a little bucket. And so these mussels never came out of the water during the course of the experiment in the field. They were always submerged, but they were cut off from the water in the ocean, or from a resupply of water in the ocean during low tide, and this pool only gets flushed and refilled really during high tide when the waves splash in it. So we have these 3 different sites here that are representative of places that mussels live in the natural environment here at Hopkins Marine Station. So there are some natural mussel beds right next to our high shore plate. So we put these things out for a total of about, 23 days during July of the last 2 years we've done this. And we go through sort of this variety of different weather conditions. So I'm just showing you some of the local scale weather conditions that these things were experiencing. In red, we've got air temperature through the day and night. Monterey is sort of notoriously cool, cold even during the summer, kind of like living up in the city in San Francisco. So the hottest days we get are about 23, 24 degrees Celsius. And a lot of time it's colder than that. Our blue line here is ocean temperature, because again, these things are getting submerged twice a day, so ocean temperature matters to them potentially quite a lot. And we had fairly warm ocean temperatures, about 16, 17 degrees Celsius during most of this time. And then at the bottom, we've got the tide cycle in black, so we went through some high tide cycles and some reduced tide cycles and some high tide cycles again. Those are the spring and neap tide cycles. And then in green is our offshore wave height, significant wave height. Basically a measure of how big the waves are crashing on these sites at high tide. So this is what the whole field site at Hopkins looked like. And then we're making our measurements at much smaller scales. So we've got all these streams of data that we can actually put together and make fun little animations of what a mussel was doing on a particular day out in the field. So what I'm going to show you here is a little sort of time-lapse recreation of a single mussel on one of those plates. The plane down here represents the tide level. So initially we're going to start out at low tide. This mussel's out in the air. Eventually the tide will rise over the course of the video, and the plane will be up here. And you'll see the mussel actually start to open up, which we have from our gape sensor. The muscle's also going to change temperature, particularly at low tide when it's out in the air, based on the data stream from our temperature sensor. And so the muscles don't actually change color in real life, but we're going to sort of mimic that with our little video here. So we start out in the early morning. It's about 9 a.m. here. The muscle starts heating up because the sun is hitting it, and it's sitting out of the water, and it's completely closed up. Eventually, the tide rises, and you see this thing starts to open up a little bit. And after an hour or two, it starts to really open up, and it's presumably sitting there feeding, pumping water inside itself, filtering out plankton, and reoxygenating itself as well. Eventually, the tide will drop again, so we'll go back to low tide. And you'll see the muscle will eventually decide that it's sort of time to maybe I need to start closing up again. Maybe I need to start conserving water inside my shells. It's actually going to heat up briefly, up to about 18, 19 degrees at the end of the afternoon when it's getting some afternoon sun. And then we get eventually to the end. We're about 8 o'clock, 9 o'clock, so the sun is set at this point in the summer. And the muscle is cooled down a bit from where it started off. So that's combining all these different streams, but then let's sort of dissect what's happening for our different groups of muscle based on those different sets of data. So we're going to start by looking at how temperature is varying among these muscles. And really this was sort of the key question I put forth at the beginning, is how much is temperature varying over small scales, and how might that be impacting performance and survival of these muscles? So I'm going to show you another little time-lapse video here. In this case, I put out a little waterproof time-lapse camera in the intertidal, sort of pointed at our two sites. And so I'm going to, in the foreground here, have the high shore plate. In the background, you can't quite see it here, is the low shore plate. And we're going to see the tide rise and then fall during the day. And I've got two plots here in the upper left and upper right, representing data from the muscles at the high shore plate and the low shore plate. And on the x-axis in each plot, I've got tide height, which you can think of as essentially a proxy for time. So the tide will rise through time and then fall through time. And then the y-axis here in both cases is temperature. And there will be a bunch of colored dots on these. The individual colored dots represent data streams from different muscles. And so as we run this, we start off in the early morning, the sun is hitting these things, and we're seeing all the muscles start to warm up. But they are warming up at different rates, even though they're just a few centimeters apart. As the tide comes in here, we see the low shore muscles cool off to ocean temperature, and eventually the high tide starts to hit the high shore plate, and you see all of the high shore muscles start to cool off. But the high tide only lasts for an hour or two or three here before it starts to drop again, and these high shore muscles will start to warm back up again as they're being just exposed to the air and no longer cooled off by the ocean. The same thing will happen later as the tide drops with the low shore muscles for just a few of the muscles there. And so really, whether you are being heated or staying fairly cool is really down to sort of your body orientation and your location in that muscle bed. Are you being shaded by your neighbors? Are you sort of under an overhang or out on top of the plate, out on top of the pile of muscles? So this is what the sort of recreation of that time series for that single day. And what I want you to take away is also the fact that there's actually quite a lot of difference between those individual muscles through time. Although when they are submerged, they are generally all about ocean temperature. So just to sort of summarize the various metrics, I mean, we've got a lot of temperature data, and what you make of it is sort of up to you. We saw maximum temperatures of muscles at the high shore and low shore of 38 and 33 degrees Celsius, but on those same days that one of the muscles was reaching 38 degrees Celsius, we had muscles that were up to 14 degrees cooler than it at the same exact time. So this range in maximum temperature that we've got here is actually quite large. And on average, on days, when we look at maximum temperatures, daily maximum temperatures, there was on average a 7-degree spread among muscles sitting just centimeters apart on the same plate. We saw maximum heating rates of upwards of 20 degrees Celsius per hour, but that only lasts for about an hour. And in general, on days with higher maximum temperatures, we also see a greater range in our plot here in terms of the difference between the hottest muscle and the coldest muscle on the plate. So some more summary data. If we look at average daily minimums, not much difference here, except I'll point out that the high shore muscles are actually getting colder, mostly at night. Again, they're sitting out in the air and they're allowed to get colder than if they were in this calm, stable ocean temperature. And over on the far right, the average daily maximum temperature, we see that the high shore mussels in red here have quite a bit of spread in terms of what their average daily maximum temperature was. Less spread in our tide pool and in our low shore plate. But it's also interesting to note that on average, the tide pool mussels were actually getting a little bit warmer because, again, they were higher on the shore and cut off from the ocean longer. And so that little pool of water they were in was actually warming up and getting them warmer on average than the low shore mussels. And then we'll just look at some summary data for within one of these plates. So this is an overhead view of our high shore plate, which was sitting essentially horizontal. And what I'm going to point out here are all of the mussels that had temperature sensors in them. Then I'm going to put up a bunch of temperature plots through time here for each of those mussels. And the temperatures that we're reporting here are average daily maximum temperatures, so the hottest these things were getting. And so you're not meant to necessarily read the details on the edge of any of these, on any of these plots, so just sort of want you to get this gestalt view of what the patterns were like. And what I want to sort of point out or highlight is that in some regions of this board, which is only the size of a cafeteria tray, we're seeing mussels that are getting up to 35 degrees Celsius, whereas their neighbors on the other edge of the board are barely reaching 25 degrees Celsius. And so there's all kinds of small-scale temperature variation happening here. At the low shore site, we see a similar thing. So this is a different plate, but we see mussels at the lower end of this plate are staying cooler than mussels sitting up near the top of this plate. And so again, lots of variation. Then let's switch into gape behavior. So this is that opening and closing of the two shell valves. I'll show you another little time lapse here, same sort of setup with our two plots and our high shore and low shore mussels. But in this case, the y-axis here is the percentage opening. So each mussel's been scaled between 0 and 100, 0 being fully closed, 100 being fully open. And again, through time or through tide height, we'll watch on the x-axis. So different colors represent different individual mussels. And early on, we start off at low tide. Everybody's closed up. They're basically sitting at 0 degrees or 0% gape. The tide will come in first on the low shore plate, and you'll see those mussels start to open up and start respiring oxygen again and hopefully feeding. And eventually the high shore plate gets washed, but it's only really being just splashed. It's never fully submerged on this particular tide. And so those mussels don't open up as wide as their low shore neighbors here. And as the tide drops again, these high shore mussels have only had an hour or two to really be open and doing oxygen exchange and recovering from their anaerobic phase, whereas the low site mussels have a much longer period of time that they get to spend open. So in general, these are the patterns that we see between those high and low shore sites. And this shouldn't be surprising, but it's interesting to sort of see the range of variation between individual mussels that are just, again, centimeters apart in terms of when they decide to open up, how widely they open, and so forth. And then let's look at inter-individual variation in gape. So again, sort of summarizing when the first mussel opened on a plate and how much or how long it stayed open versus the mussel that stayed open the least amount of time on any given day. That's what we're plotting here on the y-axis for our three sites. The tide pool and low shore mussels, we see a huge range of variation in how and when and how long those mussels spend wide open, just on that same location. Whereas the high shore mussels, which are only submerged for a small amount of time every day, we see much less variation in how long they decide to stay open. They basically open as soon as they can, and then they close as soon as that high tide has gone away, and that's it. Whereas the low tide and tide pool, low shore and tide pool mussels, some of them just decide to hang open. Maybe it's nighttime and it's cool, and they just stay open for a couple of extra 10 hours or so under those conditions. And then we can also look at sort of what time or what the pattern of opening is relative to time. So we can sort of do our comparison between time domain and frequency domain. So I've got here a time series for an individual mussel, the red lines representing how wide open it was, and you can see there are obvious changes here. And again, those are probably tied to the tide cycle here, the little inset. So we can switch into looking at spectra for gape opening or gape percentage opening data. So this is your sort of normal periodogram that you're looking at. Frequency along the x-axis, variability basically change in gape on the y-axis. Not a lot of interesting stuff happening at high frequencies here. All of the action is sort of at low frequencies or long time periods. So I want to switch basically the scaling of the axes here. So now I've converted from frequency into period because that's just more intuitive for me, sort of how long it was between major changes in gape. And then again, spectrum, or basically amount of variation that's happening at a particular frequency or a particular period. So this is for one particular muscle, going from half of our sampling rate at 2 seconds here for a 1 second sampling rate, out to a period of almost 2 weeks, sort of encompassing a good chunk of our entire deployment period. And we see two main peaks here, around 24 and 12 hours, and almost nothing happening at shorter time periods or higher frequencies. So there's no regular cyclical variation in opening and closing on the scale of, let's say, 10 minutes or 3 minutes or 30 seconds. They sort of open up and they stay open, and it really seems to be tied to tide cycle here. So I've lopped off the high frequency or low or short period and zoomed in a little bit on our sort of longer periods here. And I want to point out that the main peaks that we see here, one peak happens right under the circutidal rhythm, which is to say, or actually half circutidal rhythm, which is to say that the tide cycle is not 24 hours in a day, it's 24 hours and 50 minutes. And so the muscles seem to be peaking at about half that. And remember, we get 2 high tides a day, which should happen on average 12 hours and 50 minutes apart, or 12 hours and 25 minutes apart, instead of exactly 12 hours. So it's very clear that these muscles are making their major opening and closing movements in line with what the tide cycle is doing. And again, also at the full tide cycle period. So each line here is one of the individual muscles for our high shore plate, just to show you that they all basically line up very, very tightly on top of those same two repeat periods. The low shore site, we see again the 12-hour, 25-minute peak, and again our 24-hour and 50-minute peak. But some of these muscles do actually start to shift that second peak closer to an even 24-hour sort of day-night, light-dark cycle. And that's particularly clear, or maybe slightly clear, more clear in the tide pool site, where these muscles have started to shift that second peak over towards that 24-hour cycle. So maybe these muscles, which are always submerged, are doing some of their opening and closing based on whether it's nighttime or daytime, rather than just what the tide cycle is doing. So in general, we see similar patterns among all of these sites. And then, as I've said repeatedly, these muscles stay closed during low tides. Now, it would be possible for these muscles to open the shell valves a little bit and do some evaporative cooling if they were getting particularly hot. And that sort of behavior has been shown in a couple of other species of mussels, but it's never really been reported in this mussel that we have along the coast here, this Mytilus mussel. And so I'm here today to tell you that that's because they don't do it. So we can line up our temperature data and our gape data, here centered around time to low tide. So each line here represents a single day's trace, for a single low tide trace for one of our mussels. And I've got 3 of our hottest mussels here in these 3 plots. And color is related to temperature through time. And so when these mussels are dark, when these lines are dark red, those mussels are particularly hot, and they are staying clamped closed as much as possible. They are not doing any evaporative cooling, it appears. And so then, as I said, we also have data on body orientation from the 3-axis Excel and magnetometer. And so body orientation matters because it affects, how much of your broadside is facing the sun in the middle of the day. It also affects sort of how much access to food you might have if you're upright and higher in the flow rather than down against the rock surface and down in the boundary layer. And so we wanted to track whether the mussels were changing their orientation over the course of multiple days or weeks. So I've got a little reconstruction from one of our mussels again with our 3D model here, but we're going through much bigger jumps in time here, so across multiple days. And I've got the three sort of orientation values here, roll, pitch, and heading in our 3 different colors. And I'll just basically sum it up as saying this is one of the mussels that moved a lot over the course of our experiment. And mostly what we see is sort of a long-term change in heading, but really only if, when you look at it in 3D, it's not really actually moving that much. And mostly what it seems to be doing is just sort of rocking around on its little threaded anchor and coming to rest in different orientations as the tide goes high and low. And then this is just sort of a summary. I call this the dead bug on a windshield slide. These are polar density plots, so essentially amount of time a mussel spent pointing in a particular orientation, north, south, west, and east. And so what we see is over the course of the 3 weeks that these mussels were out, they generally stayed pointed in a particular compass direction wherever they started, essentially. We see some mussels that sort of flopped around a little bit, so a little broader spread in our direction here. But in general, we are taking this to be evidence that these mussels, once they're clamped down initially and being washed by the waves, they don't take the risk of letting those threads go and trying to reattach in a new position. They might do that over months of time, but over about 3 weeks, we don't see much difference. And then I will sort of skip over the physiological status data. You probably all don't really want to know what their osmolytes are doing and what their heat shock proteins are doing. I'll just say that we do see some relationship, particularly for our high shore mussels in red here, between how much, let's say, DNA damage they're accumulating, how much of these thermoprotectant osmolytes they're accumulating. And ultimately, the goal was, at the end of this experiment, we took all these mussels, live mussels, back into the lab, dissected out the tissues, froze them, and did these various physiological workups so that we could link their physiological status to their temperature history and their behavior history. And so mussels that were high on the shore were accumulating more DNA damage. They were also accumulating more taurine in their tissues, which is potentially a protectant osmolyte that stabilizes proteins when they're being heated up and keeps them from unfolding. So this is essentially saying these mussels on the high shore were getting hotter and also showing evidence that they were reacting to that, responding to that by building up these protectant osmolytes. And so I will go through, well, I've sort of talked about, in summary, we've seen high heating rates, a lot of range in variation in temperature, and also gaping behavior in some of the locations, but no real change in body orientation for these mussels through time. So they get locked down, and then they just sort of do what they're going to do. We see consistent patterns in valve gape that are really tightly tied to tidal cycle, even in the low shore where they're spending a lot more time submerged. They're still sort of doing their major changes in opening and closing in line with what the high and low tides are doing. And that individual mussels actually do vary quite a lot. And then something that we have to deal with as field biologists is this concept of demonic intrusion. So this is you putting out an experiment in the field and then occasionally having something come along and try to destroy your experiment for you. So I'm going to show you a short little time lapse of just about 3 minutes worth of data here for a single mussel, showing you acceleration data, so how much this thing's moving around in yellow, and the gape, how wide it is through time. I'll start this running, and initially this mussel is closed up, it's starting to get splashed, it's not moving around much, and then all of a sudden all hell's going to break loose here. And so it's going to start being accelerated upwards of 4 Gs. It's going to start opening up. And what has happened here is we have a little visitor to our experiment. So this is an oyster catcher, a bird that makes its living knocking animals off the rocks and eating the soft stuff inside. And it has seen this smorgasbord of mussels that we've laid out for it and decided to come by and eat one of them. And so it actually cracked that mussel open, shoved its beak in there, tore out all of the guts, and left us this dead instrumented mussel. And we had that happen 3 different times, actually 4 different times, to mussels on this particular plate. So I will talk very briefly about other stuff I'm doing, ongoing projects. One is trying with mussels to start monitoring heart rate. So using sensors like you get in an Apple Watch or a Fitbit to actually measure the beating or the movement of your veins, trying to do that for the hearts of mussels through the shell. And then the other thing is developing accelerometer, sort of modifying accelerometer data loggers to look at dislodgement of those algae, the plants that live in the wave-swept ocean. So we've got kelp beds out in the ocean, large kelp that grow in some cases that can be tens of meters tall, that occasionally get dislodged and washed up on the shore. They get broken by big waves. And we can model these things as fairly simple buoyant floats on a tether. And it's when, if they have enough buoyancy, they tend to stay taut and they don't do much jerking around in the waves. But as net buoyancy starts to decrease, these things can actually, their movement can become chaotic and they can occasionally be jerked out at high rates of speed and put a lot of tension on their tissue that's holding them to the rock and can break them off. And so I'm trying to develop I am developing little accelerometers that we can waterproof and attach to a piece of kelp. So this is a piece of Vogresia kelp about this tall down to the ground. Those are my feet. So we can track acceleration forces, and I've just summarized them here as net acceleration through time at different locations along the length of this thing. So in red, out at the tip, this thing is being whipped around like crazy, whereas down at the base where it's attached to the rock, in green, we've got much more gentle movement happening. And at high, sort of looking at zoomed in at a short chunk of time, again, high accelerations out on the tip, low accelerations midway and down at the base. And I will jump over these and say that partly we also want to couple this with wave measurements in situ, right around these algae. And so I've developed a submersible pressure data logger. These already existed. I just made a cheap one that's under $200 that allows us to put them out and afford to lose them occasionally, but record waves breaking right over these algae that we're measuring as well. So I will finish up there. If you are interested in any of this stuff, I am, occasionally looking for folks with sort of technical and gadgeteering ability who might be interested in working on these projects. And so please contact me at my email there. And thank a bunch of folks that have worked on the mussel project with me. Thank you.
So we have time for perhaps 3 questions.
Are these mussels, are they just for...
Sorry, do I end up doing what? So these ones we did not eat because we had to take the tissue, freeze it, and do all of our physiological workups on them. But these mussels are perfectly edible if you steam them and wash them in some butter and garlic. They make a good meal. You just have to watch out for paralytic shellfish poisoning during certain parts of the year. No big deal. You lose, the ability to move your limbs. So keep an eye out for the warning placards on the shore.
Have you noticed a trend in mussel bed density moving closer to the shoreline or closer to the shore since global temperatures have started increasing?
I think there's not really enough long-term monitoring that's able to tell us whether those sorts of patterns are happening. You can definitely go out right now and look just at a single time point and say that there are patterns in how dense the mussel beds are, low on the shore and high on the shore, probably linked to how much time they spend submerged versus exposed. But whether that's changed over long time periods, there's only 1 or 2 places in the world where I think that sort of long-term data exists. And I don't know really if there's been a major shift that way.
And also, is the growth rate with the low shore mussel bed inhabitants higher than the high shore?
Yeah, the mussels living lower on the shore, spending more time submerged and more time feeding, are definitely growing faster. You get these sort of janky little mussels living high on the shore that are probably 10 or 15 years old and have never gotten more than about 30 or 40 millimeters long. You go down a few feet lower on the shore and you'll get mussels that'll be 70, 80 millimeters long and might only be 3 or 4 years old at that point. So yeah, it makes a huge difference.
Mussels...
how do mussels react to thermal stress?
So they seem to just sort of sit there and grin and bear it. Or they don't even grin, they stay closed, right? And so they just heat up. If you're, what I would like to do, measure heart rate, you ought to see their heart rate actually increasing even while they're closed up. So they're burning energy for sure, but they're not doing much in the way of behavior during that time. Yeah.
Are there any endangered species that really don't have so much tolerance of thermal variation where we're kind of using temperature sensors in an emergency setting to determine whether we need to move populations?
I like that question. I am not... Nothing jumps to mind right now in terms of the shoreline species that are
alpine species. Maybe?
Yeah, so the alpine species. I suppose the one place where I know sort of on the shoreline is actually with abalone, which are big and make a great meal as well. And there are several endangered species, or there's one technically endangered species of abalone on our coast. And there are projects actually trying to monitor temperatures for those things to see whether they're getting too hot and getting diseased or just dying off as a result.
Okay, so it's 1 o'clock.